main thing I'm hoping to emphasize in this class is that everything is connected. So we talk about primary structure and secondary structure and tertiary structure and quaternary structure, but I want you to see how these are all interconnected, how they all at their heart involve those same sorts of interactions, how the primary sequence influences the secondary structure, which influences the tertiary structure, which influences the quaternary structure. And so although we're going to look at each of these in turn, always be thinking about how this is interconnected. But let's start. Today, we're going to talk about that secondary structure. So we've been talking about the primary structure. We've been talking about the amino acids. So remember, we have these 20 common genetically encoded proteinogenic amino acids. So those 20 main amino acids that basically are coded for by genetic instructions that tell the ribosomes what order to place them in. Remember, these amino acids all have the same generic backbone. So they have that amino group, that N-terminus, and they have the C-terminus where they have the carboxylic acid group. And then each of these has a unique side chain, or what we often call an R group. And so we've been looking at these amino acids in isolation, but now we're going to start looking at what happens when we piece them together. We're going to talk about how we can piece them together through peptide bonds. And this is going to basically give us a sequence of amino acids where he has a free N-terminus and a free C-terminus, as well as in between, you've got all these different side chains sticking off in between these amide bonds. You have that primary structure, which is your sequence of amino acids. And what's going to happen is that sequence of amino acids, it's not just going to like flop around as a big chain. Instead, what's going to happen is it's going to fold up. And it's going to fold up in a way that's basically fulfills the desires of those amino acids. And so we'll look at how the different amino acids have different desires. And so the sequence can influence the secondary structure and the tertiary structure, but also because they all have that same generic backbone, they're going to have similar desires in terms of fulfilling those generic parts, leading you to get characteristic structural units like alpha helices and beta strands and sheets, which we'll talk about a lot today. But know that this is going to be influenced by your primary structure, but it's also, you get these same structures in a lot of different proteins because that's coming from hydrogen bonding between the backbone of these amino acids. Later, we'll look at how that secondary structure, how those sheets and how those helices and things like this can team up to form different tertiary structures, where here we're actually involving the side chains, those R groups. So we get interactions between the R groups and interactions between the R groups and the backbone, and interaction between the R groups and cofactors and all sorts of cool stuff. And so that's all going to influence the kind of like overall 3D shape of the protein. And that's going to be our tertiary structure. And then some proteins will also have quaternary structure if there's multiple chains involved. And so we'll discuss that as well, how we can have things like tetramers, like in the case of hemoglobin, where you might have like 4 units of 4 chains in the 4 protein chains kind of all be working together. So that's where we're going. But today we're going to be focusing on this secondary structure. And we're going to see how the secondary structure, it's made up of interactions from the backbone, from hydrogen bonding through the backbone. And the 2 main forms we'll talk about are going to be our beta strands. So if we have these individuals, we call them beta strands. These are also been drawn as these arrows we'll look at. And then we also have our alpha helices, which are basically these helices. You also see the term beta pleated sheets. This is basically when you have multiple strands that are kind of linked together and interacting with one another. But note that a strand is going to be a single one of these, and then a sheet is when they're joined together. And then we'll also see how we can have things like beta turns, where basically they go in between those different strands to try to make the turns. And so we're going to look at how different amino acids prefer to be in a helix or a sheet or in neither. And then we'll look and see how this makes it so that different primary structures will have different secondary structures that they prefer to adopt. So that's where we're going. But first, let's back up and let's think about what we're talking about when we even talk about a peptide bond. And this is going to be really important because this peptide bond is going to have special characteristics that make it so that backbone angles that that peptide can take, that the angles that peptide backbone can take are going to be constrained to specific angles. And this is because of the unique nature of the peptide bonds. And this is going to help make it so that there's only those couple main structure, secondary structure units, those alpha helices, those beta strands. There's only a couple of main types of those, despite there being such a vast array of amino acids, because of the constrained nature of the peptide bonds. So the peptide bond, basically, it's just a name that we give. It's an amide bond. It's an amide bond, but it's in a peptide. So it's in connected amino acids. And basically what it is it's going to be, you have a carbonyl carbon attached to an oxygen, and this is going to be attached to your amino group. And so if you have 2, you get a dipeptide. You have 3, you get a tripeptide. If you have 2 amino acids join up through a single amide bond, through a single peptide bond, this is going to be an example of a dipeptide. Now, if you had 3 link up through 2 peptide bonds, that would be a tripeptide. You link up more and more, you get an oligopeptide, you link up a lot, and then you get a polypeptide. And then basically when that folds up, we call it like a functional protein. So a protein is really just like a big polypeptide, and typically it's going to be folded up in its functional to make a functional form. And the properties of that protein are going to be directly influenced by the sequence of amino acids. So what's so special about this peptide bond? Well, what's so special about it is it's resonance stabilized. So basically, we typically draw it with a double bond to the oxygen and a single bond to the nitrogen. But really, this has partial double bond character. So it's more like a 1.5 bond. And why does this matter? Well, double bonds are going to be shorter, they're going to be more stable, but they're also, in order to have that resonance, in order to have that stabilization, these are going to have to be in a plane. So it's going to be confined to a plane. So remember that resonance is basically where you have like electron delocalization in a pi system. And if you remember back to your chemistry classes when you were drawing all those orbitals and stuff, pi bonds are going to involve those p orbitals. And those p orbitals are those ones that look kind of like hourglasses, and they're going to stick up above and below the plane where you have those single bonds. And so what this means is that if you were to rotate those single bonds, what would happen is those p orbitals would get out of alignment. And if they get out of alignment, then they can't be joined. And so those atoms that are in that peptide bond, they have to be planar. And so you're going to have this constricted nature of the peptide backbone. And so there's only a couple places that you're actually going to be allowed to rotate. So this actual peptide bond, this is going to be what's called the omega angle. And so, but we're not really going to see much about the omega angle because there's only really 2 main forms that can take. It can take a cis form and it can take a trans form. And for almost all of the amino acids, we're always going to see it in the trans form. And so basically when we have the trans form, what this means, I know it sounds like I'm saying transform like Transformers, but I mean like trans space form. Basically what it means is that these C alphas, so those alpha carbons, the ones that are attached to the R group, those are going to be trans to one another. Whereas in a cis conformation, they're going to be cis to one another. So trans versus cis. And when they're cis, basically what this is going to mean is that you're going to have this R group that's going to be clashing with this carbonyl oxygen. And when you have this steric clash, it's going to be super duper uncomfy. So you're going to almost always see things in the trans form. I say almost always because there's one of the amino acids that we've learned about and that you guys will learn a lot more about, and hopefully you guys are memorizing, is your proline. So proline is basically this really awkward amino acid, and what it has is it basically has its side chain is going to kind of loop over and attach back to that backbone, nitrogen. This is going to have a couple consequences. One that's going to come into play later is going to be that it doesn't have an H attached to it. So it can't serve as a hydrogen bond donor, which is going to be important because those backbone, the hydrogen bonding to the backbone is going to be a key stabilizing force in secondary structures. And proline, well, it can't dock those. So we'll find it in things like turns, and we'll find it in other regions, but we're not going to find it in, say, a helix or a sheet or a strand. But one of the reasons why it's good for weird little tight turns is because it can form this cis form. So basically, proline is so awkward that it's always going to be causing steric clashes, whether it's in the trans form or whether it's in the cis form. And so in the trans form, you have basically the R group of the thing that's next to it is going to be clashing with the hydrogen on this proline. And note that I've just drawn in these two hydrogens here to emphasize this clash. But you're going to have those same hydrogens in the cis form, as well as there's going to be implicit hydrogens drawn throughout the structure that I'm just not drawing so that I don't clutter things. But long story short is you're going to have steric clashes in both of these forms. And so proline can form in both of these, can form both cis and trans bonds, although it's going to actually take some enzymatic help, a peptide prolyl isomerase, to actually do the switching of that omega bond to flip things, because what's going to happen is, remember, you can't just rotate around that bond freely. So instead, this enzyme is going to have to help out. But for almost all the time, for all the other amino acids, we're going to see it in that trans form. So we're not really going to think much about that omega angle, that one that's actually for the peptide bond. Instead, we're going to think about rotation being allowed at the sites around it. And so there's going to be these two main angles that we'll talk about, the phi angle and the psi angle. And so basically the phi angle, that's going to be between the nitrogen and the next alpha carbon. So this is going to be your phi angle. And then your psi angle, that's going to be between the first alpha carbon and the carbonyl carbon. So the way that I like to remember this is, I think that this psi angle, it looks kind of like a C. And then if I think, okay, carbon to carbon, you have this C. And then your nitrogen to carbon, that's where you have your phi. And I'm not going to be like, I don't need you to know, like, memorize specific angles or anything like that. And I'm going to show you, we're going to do a tutorial so you can kind of visualize things, because I think it's easiest to see this in 3D. But I just want you to know that basically there are these two main locations where you can rotate, but most places you can't rotate. Right here, we're just looking at a dipeptide, a single peptide bond. But basically what's going to happen is that when these link up, you're going to kind of get a chain of planes where you can only get the rotations at those specific points. And when we have these phi and these psi angles, these are what we call dihedral angles. And that sounds like a scary term, but basically it just means angle between two planes. So basically, you'll see we have this chain of plane, and then we're going to have these places where we can turn, and we can talk about them in terms of their dihedral angles. Your book, as well as other places, you'll see it kind of like drawn out in this projection where you're going to be looking down the bond at the C alphas, like in front of one another, and then looking to see as you turn things, as you introduce like torsional strain as well as steric strain. But I'm going to have you look mainly through those 3D tutorials and interactions that I'll show you in a minute, because I think that those are going to be the easiest way to really see what's going on. And what you're going to see is that as you turn those, as you rotate things around those phi and psi angles, a lot of places, most of the places, you're going to get significant clashes, you're going to get significant strain. And there's only going to be a couple of places that things are really favored. And these are going to correspond to those characteristic secondary structures that we're going to see. So let's go take a look. So there's a tutorial as well as an interactive version that you can go and actually play with things yourself, which I highly recommend that you do. Basically, there's really great tool where you can highlight things, like you can highlight the phi and the psi angles, as well as the planes and the peptide bonds, You can also show the van der Waals. So basically show the space that these atoms will actually fill. So it makes it easier to see the clashes. And then you can also explicitly show those clashes. And so what you'll see is that as you change the dihedral angles, you're going to introduce clashes or you're going to not introduce clashes. And this is going to help you see that there are only specific angles that these dihedral angles can take without causing these steric clashes. And this is going to be one of the main reasons why we have the backbone structure limited to those particular angles. Now, when we have those angles, we can actually go ahead and we can plot those angles on this plot called a Ramachandran plot. And so I'm not going to make you know like what's where in one of these plots, or to be able to know the exact angles that an alpha helix or a beta strand will take. Just know that there are distinct angles that they prefer to take. And so if you were to go and you were to plot out the angles that these different atoms take in all the different protein structures that have been solved, so all those extracrystallography structures and stuff, and you were to take them and you were to plot out the points, the phi and the psi angles for each of them, what you'd see is that they cluster in different regions in this plot. And where they cluster the most, that would be like a highly favorable region. And where there's few of them, that would be a disfavorable region. And so we'll look at a little more detail about this. But this is one of the tools that structural biologists use in order to know if their structure seems reasonable. You look for like what's called Ramachandran outliers, which are points that are outside of these like preferred ranges, which might tell you that there's something going on wrong with your model there. Or it might tell you that there's a really flexible part or a really weird part of the protein, which might actually have functional relevance. And as we'll see, glycine and proline are going to kind of fall outside of these often. But there are typical places where you'll see angles, basically the points cluster, corresponding to various types of secondary structure, because those various secondary structures have characteristic phi and psi angles. There is another tool that you can use. There's actually a couple other really nice tools, and they're going to let you actually like go ahead and stick in the phi and psi angles and see where it corresponds on the Ramachandran plot directly. Okay, so now we hopefully have a better idea of kind of what these phi and psi angles are, as well as how they're represented on a Ramachandran plot. So remember that those dark regions are going to be like the core, where things are most often found. And so there will be places where you're most often to find beta strands, places you're most often to find left-handed alpha helices and right-handed alpha helices. You'll see that the right-handed alpha helices, these are going to be much more common than your left-handed. And left-handed, you'll only really see in places like weird structures like the structure of collagen, which also has this weird triple helix, which is really, really cool. So maybe if we have time, we'll get to it later. But most of the time, what we're going to be dealing with is a right-handed alpha helix. So note that when we talk about the handedness of a helix, basically what you want to do is you want to take your thumb and give a thumbs up and kind of rotate your thumb as you give that thumbs up, rotate your wrist as you give that thumbs up. And if you're doing it with your right hand, you should be tracing the right-handed alpha helix. If you were to do it with your left hand, that would be tracing a left-handed alpha helix. So when we talk about alpha helixes, in the case of structures, usually these are going to be right-handed. And these are going to be really, really common. So about a quarter of amino acid, about a quarter of like proteins are typically in this alpha helix form. And we'll talk more about it in a minute. But that's what we're typically going to be seeing. And then we'll see that this is going to correspond to a specific region on the Ramachandran plot. And I'm not going to make you know which regions which, just know that they do correspond to a specific region because they do have these specific preferred angles. But we also have this big contribution of these beta strands. And so we'll also talk about what makes a beta strand and why these angles are different than those of an alpha helix and why different amino acids might prefer to be in different places. But remember that the dark regions where you have a lot of dots clustered, those are going to be the most preferred regions. Then the lighter regions, they're like, okay, well, that's kind of okay. If you're outside of those regions, then something might be weird with your structure. except if you're looking at glycine and proline. So often when you're looking at a Ramachandran plot, say for a structural model, it'll say like non-gly, non-pro Ramachandran plot, and then it'll be like a separate one for glycine or proline. And this is because these guys are weird. So I already mentioned that proline was weird because it had that really awkward backbone contortions. And then when it's linked up, you're not even going to have this hydrogen here, so it can't form the hydrogen bonding. In the case of glycine, well, its side chain is really small and really flexible. And so basically it's going to be able to take on a lot more different angles. So what's going to happen is that if you were to look, you would see that glycine, it could be in a lot more different places, and proline, it's going to be in a more constricted places. But you'll also see it in places you want to see other ones because it can take on some kind of awkward angles, but constrained awkward angles. You also see, however, that although those are kind of like our extremes, we'll also see that the size of amino acids in general can influence the angles that they can take because of that steric hindrance, because of these amino acids needing space, like they need a little elbow room. And so if you have big bulky things next to each other, what's going to happen is that even if you're in those like typically preferred angles, if those R groups are next to each other, they're going to be clashing. And so these things might be better off in, say, a beta strand, as we'll look at, because there the side chains are kind of be going up and down, alternating, whereas in a helix, they're going to be next to one another. And so we'll look at this in a minute. Just know that the size of the side chain is also going to influence the angles that things can take, providing yet another link between our primary structure and our secondary structure. So now let's dive deeper into that secondary structure. So the two main structural units we're going to talk about are beta strands, which can team up into beta pleated sheets. And we'll talk about how these sheets, basically, this is where you have multiple strands interacting with one another. And these can be in a parallel manner where the strands are going in the same direction, or they can be in an anti-parallel manner where they're going in opposite direction. And then we'll also talk about alpha helices, which are going to be these helix-looking things, which again are going to often typically be in your right-handed form, as opposed to DNA, which is typically going to be in a left-handed helical form. So we'll look in detail about how different hydrogen bonds interactions are going to be stabilizing the alpha helix and the beta strands in the sheets. But first, I just wanted to go tell you a little bit more about these different ribbon diagram models, where we show these beta strands as kind of these flat arrows that look kind of like, I don't know, what are those pasta, those thick noodles, you know, it looks kind of like that, but then with an arrow at the end. And basically the arrow is going to be pointing in the direction of kind of the protein. So remember that proteins are going to go from N to C. So when you have one of these arrows, basically what it's going to be saying is that this is from N to, and then this is from C. So basically you're going from the N to the C. So for each of these strands, you could think of it as N to C, N to C, N to C. But basically overall, this is going to be really your N terminus, and this is really going to be your C terminus. And this is just talking about within this region, it's going in the N to C direction. But all of this is going to be one chain. And you'll see that in order for these strands to link up, what's going to happen is that you're going to have some sort of more flexible loopy region in between them. And so we'll look at how we can have these tight turns. So this would be an example of a beta turn. And we'll look more about what makes a good beta turn, as well as we can have more flexible regions. And so if you have these like parallel beta sheets, these have to actually have a longer linker region, and they can actually involve like strands from regions of a protein that are pretty separated in terms of their actual sequence, because it might not just be a single linker region. It might be two strands from totally different places on the peptide sequence that are kind of interacting with one another. So that's one of the main reasons I like to think about secondary structure in terms of defining it as kind of being involving the backbone interactions. Because what happens is, like, sometimes you'll see it defined as like local structural units, but sometimes these beta strands can be involving regions that are actually pretty spatially distant in their primary sequence, but close in their actual 3D space. In terms of these actual diagrams, though, in terms of these flat noodles, which someone needs to in class remind me what these things are called, and these kind of like tubey looking helices, these ribbons, This is called a ribbon diagram, and the inventor of this representation is this really amazing scientist named Jane Richardson, who I've had the pleasure of meeting in person several times at Cold Spring Harbor, where I did my PhD, and I sat in on the X-ray crystallography course a couple times, and she taught lessons there, and she's really, really amazing. And she came up with this structural model. So in this class, we're going to look at a bunch of different ways that we can represent the 3D structure of proteins, and that we're going to see that they're going to emphasize different features. And so when we talk about the ribbon diagram representation, this is going to be emphasizing the secondary structure. So it's going to make it easier to see the alpha helices, to see these beta strands. We're typically not going to be showing like the side chains and things like this when we have these, but we are going to be able to look and see the secondary structure, the overall structure of the protein. And then if we want the nitty gritty details of the individual amino acids and things, then we can do things like use a stick representation. One of the reasons why we might want to use, say, a stick representation, so such as this would be a stick, and this would be space filling. One reason we might want to use a stick model is so that we can better see where we have things like hydrogen bonding interactions, because those hydrogen bonding interactions of the backbone are what are actually going to be stabilizing these secondary structural motifs. When we're talking about an alpha helix, we're involving hydrogen bonding between the backbone oxygens and the backbone nitrogens, the hydrogens. So that'll be the hydrogens of the backbone nitrogens, 4 residues later. So we're going to have N plus 4 are going to be interacting. So we'll have N, and then the N plus 4 residue are going to be interacting. So your oxygen here, and then your hydrogen on the nitrogen would be your N plus 4. And so these are going to be what's actually interacting. A really important thing to note is that the side chains themselves are sticking out. So side chains stick out. So they're all sticking away from the helix. And then although there looks like there's a hole in the middle of the helix, there's really not. If you look at the space filling model, it's going to be like all filled up pretty much. So, but we have the side chains sticking out. And why this matters is because they're all sticking out in the same direction. So they're all going to be next to one another. And if you have big bulky groups next to one another, well, now you're going to have the opportunity for there to be steric clashing. And so you can get steric clashing between the side chains if they're big and bulky. So we don't want big and bulky. What would we want? What would be good for an alpha helix? Well, something that would be good for an alpha helix, well, now we want something that's going to be long. We want something that's going to be flexible. We want something that doesn't have its bulk close to the backbone, yeah. And so we'll look and we'll see that you find different amino acids will have different favorabilities in an alpha helix, and we won't really find those big bulky things. We won't find those things with their bulk close to the backbone in an alpha helix, because then those are going to be clashing with one another. And instead, we're going to see lots of these long, flexible ones. And we will see charged things and we'll see hydrophobic things. And sometimes we'll see the charged things will be on one side of the alpha helix facing the solvent. And then the other side will be out in the hydrophobic things that are going to be facing the interior of the protein. And we'll see this in our exercise in class. But one thing that you don't want is you don't want things that are charged next to one another. So you don't want neighboring same charge, because that's going to cause repulsion. And we don't want big and bulky, and we also don't want really, really tiny. So we don't want glycine, and we don't want proline. Remember, proline can't form those hydrogen bonds. Glycine, it's too small, it's too flexible, it's going to destabilize things. So we don't want either of those in an alpha helix. It's not typically like one amino acid is like always in one form, and that if you have that amino acid, boom, you've got a helix. Instead, if you look at a sequence and you keep kind of a string of amino acids that all have a high propensity to form an alpha helix, then it's likely something that does form an alpha helix. And if you look at something and you see it's got glycines and prolines and all that stuff, that would be something that would not be forming an alpha helix. And if you see something with a bunch of big bulky groups, that also probably wouldn't be forming an alpha helix. But that might be forming one of the beta strands, because in the beta strands, well, here it's okay to be bulky, because here your side chains are going to be sticking up and down. They're going to be sticking away from one another. So we can say big bulky, no problem. So that's going to be okay. But it's still not going to be okay to have your glycine or your proline. Those poor guys aren't, they can't be anywhere, pretty much in terms of these secondary structures, but they're good to be in these turns. So they can be in these like beta turns. And so those two main beta turns I will look at, and these are going to be those different types. They can often contain these glycine and these proline, which can make those nice tight curves. So remember, the strands are the individual units, and then those sheets are when they kind of link up. And we call them beta pleated sheets often because we got this zigzag structure. So we have this zigzag structure, and what's important is that the side chains go above and below. Above and below this zigzag, so this like pleated strands. And then what's going to happen is that these strands, the actual hydrogen bonding is coming in between these different strands. So it's not in between a single strand, but it's in between multiple strands that are pairing up to form these sheets. And again, these can be parallel sheets or they can be anti-parallel sheets. So if you have something that is anti-parallel, well, here it's easy for the strands to kind of come together to form these sheets. You only need this tight little turn in between them. But if your strands are anti-parallel, well, now you're going to have some sort of bigger connecting region, and this can actually be uniting strands from very different places in the primary sequence, in the primary structure, the sequence of amino acids. In both of these cases, what you're going to see is you're going to get hydrogen bonding between those oxygens and the hydrogens of the nitrogens of different strands. So you're seeing hydrogen bonding between the different strands, whereas in the case of the alpha helix, we're seeing bonding between the atoms in the same strand. But with these beta sheets, they're between different strands. Also remember that in your alpha helix, basically the things are sticking out. And in the case of your beta sheets and your beta strands, things are sticking up and down. And when I say things, I'm talking about those side chains. And so now let's look at how this is actually going to influence what sort of structures different amino acids will adopt. Well, if we start from the very basics, if we're just thinking about those phi and psi angles and rotating those, we can already kind of see that the size of the amino acid is going to influence things. So we talked about glycine. Yeah, it's really small. It's really flexible. So we can see it in different places. But what about things on the other side of the other extreme? What about things like tryptophan? What about things like tyrosine? When you rotate those, they're going to kind of like clash with things. Even things that don't look that big, things like when we have our threonine, it doesn't look that big, right? But look, its bulk is really close to its backbone. And so it's going to be close in that area that's going to then make it clash with the things that are next to each other. If it's next to another R group, like directly, such as in an alpha helix, well, that can actually clash. But if it's kind of on opposite sides of something, such as we have in a beta strand, well, that might be okay. So maybe we'd expect something like a threonine to be in a beta strand. And maybe we'd expect something that was like long and flexible, something maybe like a lysine to be in an alpha helix, because, well, now it can kind of spread its bulk away from the backbone where it doesn't have a chance to interfere with anything around it. So that's what we would kind of think, right? But what do the numbers say? So there's actually scientists have looked into this, and they've actually done it a couple of different ways. You do see some concurrence. And again, these aren't strict rules. And what I really care about is that you kind of get the trends. We're seeing that we have long, flexible things in an alpha helix. We don't want big, bulky things. Don't want big, bulky things next to each other. because remember, all those side chains are all facing out, so they don't have that benefit of being spaced out above and below. Instead, they're all like next to the one that's actually adjacent to them. And they're also, remember, they're going to be able to interact with the one that's 3 or 4 away from them. We don't want there to be unfavorable interactions with the ones that are kind of above one another in the helix, as well as the ones that are next to one another in the helix. Less of an issue in the case of our beta strand, where you have things alternating above and below. In either of these cases, you can kind of think about there being different faces. And so we'll look at this when we do our modeling, but basically you can think about, you can have this one side and you can have this other side. We talk about sides, but I mean, it's a circle, so you can be having lots of sides, like an infinite number of sides. For your beta strands, though, there really will be this one side and there'll be this other side. And so you might see alternating properties of things, like maybe you have a hydrophilic thing and a hydrophobic thing, and then a hydrophilic thing, and then a hydrophobic thing, because they're alternating up and down. So I don't need you guys to remember the exact number of residues, like the exact angstrom distances or anything like that. You don't need to know the exact values of the phi and psi angles, but know that those all come down to the restrictions based on the peptide bond, and based on the optimal angles that would allow them to form the most favorable hydrogen bonds, the most number of hydrogen bonds. It's all about that. That's what it comes down to. The angles are constricted by those, by the resonance of the planar peptide bonds to specific angles. So specific angles are then further refined based on the requirements for wanting to meet the hydrogen bond donors. And all of that is going to be affected by the primary sequence of the amino acid, where the size of that and where that size is located in is going to impact the actual angles it can take without clashing with things next to it. And the charge and things of those amino acids might also influence whether they want to be next to each other, whether they want to be 4 apart from one another. So the primary sequence is really of primary importance for determining secondary structure. And there are tools that can actually predict the secondary and even the tertiary structure based just on the primary structure. Things like AlphaFold and Rosetta Fold and various other machine learning things that you probably have heard about in the news. But really, it all comes down to the biochemistry. And although we've been talking about all these secondary structures, it's also important to realize that not all regions of a protein actually have a defined secondary structure. And there's these things called IDRs, or intrinsically disordered regions. And these could be just like a small part of a protein, or they could be like most of the protein in some cases. But these don't really have set structures. They're more loosey-goosey, but that doesn't mean they're not important. They can actually serve great roles for scaffolding by bringing multiple things together. Sometimes they actually adopt the secondary structure, set secondary structure once they bind things. So very cool stuff. They can also do things like phase separation, where basically they bring stuff together and make things form this gel. So you can kind of get these membraneless compartments within cells. So IDRs are going to be another crucial thing. They by definition don't have a set secondary structure. And often in these regions, you're going to see things that are kind of going to be really hydrophilic. You often see charged things as well. Various things, they're often interacting with RNA and DNA. So you often see positively charged things that are going to interact with these negatively charged nucleic acids, various things like that. As to how scientists actually look and kind of study secondary structure, there are a few different ways. So the main ways are just going to be our common structural biology techniques, our X-ray crystallography, and our cryo-EM. That's how we're getting those ribbon diagrams. That's how we're getting all those stick diagrams and all those space filling diagrams, which are all just different representations of the same structural model that was formed from that data collected through these experimental methods. There's also a lower resolution method called circular dichroism, which is a lot easier to do. And basically it tells you about various secondary structure, the proportion of your helices, the proportion of your beta strands, the proportion that's kind of unstructured. But it's not going to tell you what region is what. It'll just kind of give you an idea about the overall conformation. And so you won't need to know how this technique works or anything like that. I just wanted you to know that it does exist, and it involves how these different structures interact differently with light. So hopefully this helps you see that that primary structure, that sequence of amino acids, is going to directly influence which secondary structures things will take on. And then later we'll look and see how the secondary structures can combine to give us things like our tertiary structure and our quaternary structure.